I'm the first talk in the bioinformatics track, so this is kind of exciting. For everybody who's all hyped to go learn about DNA sequencing or other things, I'm sorry, we're going to talk about flow cytometry instead. My name is Jacob Frelinger. I'm from Duke University in the program of computational biology and bioinformatics. And I work in a computational immunology lab. There we go. I'm going to talk a little bit about the background of what is flow cytometry, on the assumption that most of you don't actually do flow, and how to go through and do a flow analysis and introduce how to do it using this library I've written, FCM. And we're going to cover traditional gating-based analysis and these new quantitative model-based methods that we've been developing. And then a couple other features of the library that I've included, and then sort of the future direction of where I think FCM needs to go. Just get some boilerplate out of the way, so if anybody ends up really interested and downloads the talk later, the links are there. And our dependencies, we've tried to keep it very simple. We depend on NumPy, SciPy, and Matplotlib, which you all should have. Our model-based methods are based on a library coming out of the stats department at Duke called DPMix we've been involved with developing. And we've also leveraged the GPU stats library Wes McKinney helped write to try and get us some speed-ups in some of our computations in the big and in the models as well. So, Why are people interested in flow cytometry? It's one of the oldest high throughput methods in biology. It really came to prominence in the mid-'80s with 3 and 4 color flow cytometers becoming available. It's unique, it's multi-parametric, meaning you get lots of parameters at a single cell level from heterogeneous samples. Typically it's just peripheral blood for the work I use. So you have a whole mix of different types of cells in there. So how flow cytometry works is that cells are labeled, typically with fluorescently coupled monoclonal antibodies targeting specific proteins or cell receptors. They are stained with these. Each of these different markers has it's a unique fluorescent tag associated with it. It is then fed through a detection device past a laser, which causes the fluorescent molecules to excite, emit fluorescent light. And then that's recorded and onto a computer and generates a binary file called an FCS file. Once you have this, the goal then typically is to try and identify specific cellular populations by examining their fluorescence properties, which correlates then to quantities of surface proteins or whatever the antibody was tagged to. So in this case, we're looking at, on the x-axis, the CD3 fluorescence intensity. So cells to the right exhibit more the CD3 cell surface receptor. In addition to recording that, as they pass by the lasers, it measures also a forward scatter and a side scatter of the deflection of the laser beam, which roughly corresponds then to the size of the cell for the forward scatter and the complexity of the cell for the side scatter. So your typical workflow after you've run your sample through the machine is you will open up the file, you will do some quality control, do some plotting of the data, and then going through gating, which I'll describe later, you try and identify these single cell population, or these purified cell populations, and record some summary statistic, typically either mean fluorescence intensity of a particular marker, or more commonly the frequency compared to some parent population, such as frequency of activated T cells versus total T cells. And after you've done that, you generate those statistics and do some reporting based on that. So FCM has a load FCS function, and pretty straightforward. Just pass in either a string or a file handle to an FCS file. We support, right now, FCS2, FCS3, and FCS 3.1 files. Older versions of FCS files, I'm not sure, because earlier versions of the file standard weren't very clear, and oftentimes contain analog data that's basically, I believe, unrecoverable at this point. They embedded raw histograms in the file, which is probably unreadable by just about anything at this point. But modern flow cytometers will all give out FCS 2 or 3 files, and you can load them, which will then return what we call an FCM data object that represents all the information in that file. Once you've loaded the file, you'll typically have to do compensation. Because of the fluorescence properties, that your fluorescent colors will bleed over into neighboring colors. You need to account for this and then try and remove it. As you can see here, if FL1 is our green color, FL2 is our red, there is a significant amount of green in our red signal. Most flow cytometers now will allow you to produce compensation matrices, which then you can then use to remove this spillover. And given then, if you have a compensation matrix and what laser colors it's associated each channel of that with, you can then apply compensation at load time, or you can apply it later. The metadata associated with an FCS file, inside the FCS file, often has a spillover matrix. And if you don't ask for anything in particular, it will just apply that one if it's there. Another thing to view data, flow data is assumed to be log normal for most parameters. So to visualize, as you can see on the leftmost graph is our untransformed data. Typically, people will then apply log or log-like transforms, the most popular being called logical. That's a mix of sort of linear and log transform to try and deal with some of the artifacting that log transform can't handle negative values and zeros very well. So it attempts to sort of be linear around that. And it's sort of the more popular transform these days. So you can specify the transform you want at load time, just to make things easier when loading files, or you can do it on your own. FCM tries to be intelligent. If you don't specify a transform, if it sees data scaled in the newer format files that almost all then want logical transform, it will default to that. Otherwise, it does nothing. So as I said earlier, there's metadata in the binary file you get out of the machine. It often contains additional information, such as what the laser configuration was, what antibodies are associated with each color. And so you still have access to all of that. Most of the open source tools I've seen for flow cytometry simply just take an FCS file and dump out CSV and throw out all this additional metadata. We kept all of it in there, including things you probably would be totally uninterested in, like where the offsets in the file were, where to start reading data and metadata and things like that. There's also an analysis segment in the FCS standard. I've never seen it used, but we have it in case it ever shows up. And then once you have loaded your FCS file, if you wanted to just get started playing with it, we treat it almost like we've built it as a wraparound in NumPy array. And so we delegate to NumPy for most of the common things you'd used to. So you can ask for its shape, you can call its mean, its standard deviation. You can pass it to just about every function that takes a NumPy array. I haven't run into it, doesn't work. Or if you wanted to start getting out the fluorescence values or the scatter values for different channels, you can just start indexing it like a NumPy array. I also wrote in a method so you don't have to keep track of what channel is associated with what antibody. And so if you can ask for them by their name as well, which is specified in the header. And since we delegate to NumPy, you can just plot like you would most any array. Just load the file and then call scatter, and it'll plot happily away. We have included a handful of sort of graphics convenience functions. Typically, when the data is logical transform, the flow cytometrists who do the analysis normally really like to see the scale done in the sort of log-like transform. And so we've written some convenience functions to set the x and y axes to that scaling for you, so it matches the data. But since you can plot just like any NumPy array, you can create crazy plots like this. So traditional analysis, once you have your data loaded, is to identify specific cellular populations. And that's typically done by gating, which is in some form of drawing some boundary in which you then take cells within the boundary and then typically perform another projection in a different set of parameters to slowly gate down to your purified population. So as we said before, this is grabbing out all the CD3 T cells then, since CD3 is the T cell marker. And so you do it iteratively, and so this is sort of a typical analysis we did to find, I found activated CD8 T cells. So gating through, we grab, gates would then be named singlet gates. Those are viable CD3, then CD8, and then the bottom three on the left are all cytokine markers, which we're using as a proxy for activation. And then we plotted them to look at their maturational states. And therefore, this population down here would be sort of one of our end target goal populations to find out how many cells there were. And so in a typical experiment, we'd look at multiple of these and try and decide if treatment had more activated T cells or had more memory T cells than case and control. A typical file will have hundreds of thousands. So we deal mostly in clinical, so ours go from 100,000 to 500,000, but millions are possible. Our sample little toy one for development was 10,000 events. But they also are often high dimensional in that, we currently cutting edge fluorescent flow cytometry can do 18 parameters. There are new ones coming online based on heavy metal isotopes instead of fluorescents that currently can do 30 parameters, and they believe they can push that up to 60 quickly. They're just slowed down by they have to create then these monoclonal antibodies with the heavy metal tags. But I've seen 30 parameter data. So this hand drawing of gates is got to go away because this takes forever. A typical clinical sample is 20 patients, 4 or 5 samples each. So we support right now most of the common gates I've seen used. The standard, most common one you'll see is a polygon, just a set of vertices defining a region in space. And then a quadrant, which basically takes a 2D projection and divides it into 4 quadrants. Thresholds, which is all events above a certain value, and an interval, which is all points between 2 values. And going through, as we've seen, this is representative of polygon. This is quadrant. This is a threshold. And you can define gates in FCM. I've just defined a really simple polygon gate here. It's 4 points. And then the second set is the axes to gate on. I just actually found a bug. You're supposed to be able to use the names. Again, didn't work, so I had to fix that. And then you can just apply the gate to the data object. One thing of note here is we store all of the analysis in the FCM data object. As you gate, you build up a tree. You can move up and down of each population as you go through. So that's what this data.visit root and data.visit G1 is. So we visit the original population and we're plotting it in gray. And then we visit into our gated population and then plot it in blue to get you a picture like this. So manual gating-based analysis. Oh, wow, all right. Got to speed things up because this is the interesting part. Model-based analysis. Traditional gating-based flow doesn't scale well. The more parameters means you got to figure out what plots you're going to look at, how to find your populations. Doesn't scale well, near impossible to reproduce. Takes lots of training to get things close. Doesn't work. So there's been big interest in sort of bringing mathematical tools online to try and automate the identification of populations for you. And they're popular because they're data-driven, the right models, they scale well with dimension. And so we implement basically 3 models, well, 1 and a variation, 2 and a variation. We have a simple k-means, just does standard k-means clustering. And another one based on a Dirichlet process mixture model, a mixture of Gaussians, and then a hierarchical one which simply fits across multiple data sets at the same time. We are just using SciPy's k-means. So we don't use k-means often. We just implemented it because that was sort of, at the time we started, the popular one in the literature when model-based analysis was just getting off the ground. Yeah, well, it is. So yes, it takes lots of training to know that if you want activated, memory T cells, you have to look at CD3 positive events, then either CD4 or CD8 positive events, but exclusively, right? Because otherwise it's a double positive. And then, so there's lots of biological information to go into producing that. Let's get back here. So mixture models, we treat it as the data is coming from a probability distribution comprised of lots of simple distributions that then sum up to create the more complex distribution. And for our mixture models, we use a mixture of Gaussians, which means we just say for k Gaussians, the probability of observing x, if you know the actual weight of each distribution and the mean and the covariance, is just a straightforward calculation then. We provide 2 methods for estimating the underlying distribution. 2 minutes, okay. One using Markov chain Monte Carlo and another one using Bayesian expectation maximization. And we've tried to include sensible default hyperparameters for all our stuff so that simply to fit data, you just instantiate the model, pass in the data to fit, you get a results object out, which then you can use to classify, predict different data sets, calculate the probabilities. And if you classify the data, you can then print by cluster assignment then. Sort of burning through this. All right, some other features we have. We worked on an information measure to help design panels and to identify better gating strategies. Since we work with clinical data a lot, there was a lot of interest in how can we distribute anonymized data, and we ended up writing an FCS writer so that you could go through and edit the metadata associated with an FCS file and then write it out. And because we work with our flow clinical lab is based around FlowJo, which is a proprietary software for doing flow cytometry, we've written some compatibility stuff to load FlowJo workspaces and extract the compensation matrices and gates out of it. And a postdoc in our group has been writing a GUI-based application to try and help biologists then do model-based analysis easier without having to do this. I'll skip that. Thanks to my advisor, Clibran Chan, and Adam Richards, who's a postdoc in our group writing Cytostream, the statistics department at Duke, who's been helpful in working out all the details on the models, Mike West's group in particular, Andrew Cron and Lynn Lin is two of his grad students, and the Center for AIDS Research at Duke, who has been our flow collaborators, run by Kent Weinhold and the Janet Stats. Questions? well, yeah.